Welcome to Sigest, your fortnightly science from the Bioeconomy Science Institute. Kia ora koutou katoa. Welcome to Sigest. I'm your host, Hilary Ireland. A strange-looking weed harming livestock, scabby skin lesions on an expensive shipment of fruit, sick and dying kauri, and oh dear, is that a brown marmorated stink bug? When faced with threats to biosecurity and when poised with critical research questions, people turn to Aotearoa New Zealand's nationally significant collections and databases for resources and expertise. Today we speak with Dr Bevan Weir, Research Priority Leader for Collections, Databases and Curation at the Bioeconomy Science Institute. We'll learn about the impact some of these collections have had in supporting research and biosecurity efforts in Aotearoa New Zealand. Dr Bevan Weir, welcome to the podcast.
Great, thank you. I'm happy to be here and share my passion for the collections and databases.
Great. Before we learn more about the collections, can you tell us a bit about yourself, such as your field of expertise and how long you've been in the field?
I started 24 years ago and I did a PhD in microbiology, and I was looking at the bacteria that live in the roots of our native legumes like kauri and kakabik, and also some of the exotic weedy legumes like acacia, the wattles, and gorse and broom. And then I moved on and I did a little bit of contracting work for a while, working for MPI, and I even had fun working for Corbyn's Viticulture out in the vineyards. And then sort of came back to core science doing my postdoc on a fungus called Coleototricum, which is a very serious pathogen of many crops. And now I lead the collections and databases.
Wonderful. Gosh, that sounds like a really varied career. So what are nationally significant collections and databases? What purpose do they serve?
Yeah, so the nationally significant collections and databases were set up by the government in 1992, just after DSIR broke up into the different organisations. And it was recognised that these collections have existed for many decades prior, and they were so important that they needed to be continually funded in the future, and that the normal mechanisms, how we fund science and sort of asking for money and putting in bids and getting funded or not, just wouldn't really work for these assets that we need to keep for really, really long term. So they decided to scour the country and look for the most important collections and databases that fit this criteria, that they were unique, special, didn't exist anywhere else in the world, and that they needed to be continually funded at some level to maintain their existence. And some of these in other organisations you might be familiar with, like GeoNet, that is the earthquake database. And in the BSI now we have 11 of these nationally significant collections.
Okay, great. Yes, so the Bioeconomy Science Institute has a range, 11 of these collections and databases, and covered a few. We have a few including several germplasm collections for crop and pasture breeding, soil and land cover databases, vegetative structure databases, and many others of significance to New Zealand. But today we're going to learn about the collections that help create impact for biodiversity and biosecurity. So maybe before we start this, given that the science of taxonomy is so fundamental to these collections, can you tell us about the science of taxonomy and maybe how correct identification of an organism influences approaches to biosecurity and biodiversity?
Sure. Well, I just found out last night that the science of taxonomy turns 300 years old in 10 years' time. So it's a very old science. It's an important one because it determines the names of organisms that we have. And that might seem kind of trivial if you're thinking about big things, like you can look at a kiwi and you know, oh, that's a kiwi, or a lion or a tiger, all these really large, obvious organisms, kauri trees, for example. But taxonomy goes across all of the aspects of life, including the little microbes and other organisms as well. And there, it's really important that we have rules about how these organisms are named and how they are classified. And because this is really, really old science, so 300 years old, how organisms are named comes from that time. And so everything has a Latin name. So humans, for example, Homo sapiens is the Latin binomial, so the genus and the species name. And taxonomy is the science of describing new organisms and deciding what the species boundaries are. So within kiwi, for example, what makes brown kiwi different from the other species? And in the biosecurity field, of course, having a correct name is really, really important, because if we're making a decision at the border to stop something coming in or not, or if this is going to be a problem species or not to control, we need a very, very accurate name and understanding of what that species is. An example of where this was important was a few years ago when I was working on kauri dieback disease. And so we all know kauri trees are incredibly important species in the northern forests. They can, be enormous and live for more than 1,000 years. But in 2006, we noticed that there was a lot of kauri trees dying in the Waitakere ranges. And we tracked it down and found that it was a certain pathogen called Phytophthora. But the taxonomy comes in it when we were trying to figure out, well, what is this species, and is it a new thing, or had it been here for a while? And we were able to use the collection to track back a sample from the early 1970s on Great Barrier Island, and with the taxonomy compare it to a bunch of other species around the world, and we discovered that it was something new. Nobody had ever seen this before, which has meant that we needed to give it a name. And of course, 300-year-old science, and so I had to give it a Latin name, and in Latin it's Phytophthora agathidicida, which is a little bit of a mouthful, but if we translate that into English, it means the plant-destroying kauri killer.
Gosh, so you named that yourself?
Yes, I did.
That's exciting. How long did it take you to come up with that?
It was probably about 5 years' work putting it all together. It was a bit of a complicated story, and we had some somewhat similar species in the Pacific that cause disease on coconut. And so we did a lot of work with both genetics and microscopy, looking at all the spores and the structures to confirm that it was indeed a new species.
So which collection would that have been utilising?
So this was using the ICMP, or the International Collection of Microorganisms from Plants. This is a collection of microbes, I guess, different from many of the other collections in that it's alive. So all of these bacteria and fungal samples, and we've got about 24,000 of them, are all kept alive but frozen in liquid nitrogen. So that means we can retrieve them and bring them back to life from their frozen state. And really importantly, being alive, we can do experiments, and so we can then test them. So what plant does this organism cause a disease on or not?
And so of all of the related species to the kauri dieback disease, what was so special about this one that, created this disease for kauri?
In terms of its morphology, it was pretty standard, similar to these other species. So the other species was this coconut pathogen in the Pacific, a pathogen of chestnut trees in Asia, and then another more globally distributed species. The interesting thing is, where did this come from? So, kauri are thousands of years old, and they've been living quite happily for all that time. So with pathogens and their hosts, there's always some kind of balance. And so the pathogen can't completely kill the host, because then it's got nothing left to infect the next time. And so these kauri disease organisms probably came from another similar tree in the Pacific.
Right, okay. So kauri dieback is certainly a story that many, if not all, New Zealanders know about. How have some of the other collections influenced biodiversity research and efforts in conservation here in New Zealand?
The biodiversity aspect of the collections is fascinating. So I'm a nature lover. I love, walking out in the forest, the ngahere, and seeing all the different organisms. And I've recently got into bird photography, so getting out and taking photos of birds. And so one of the fascinating things for me about all of the collections is the incredible diversity that you just wouldn't know exists. So, for example, we all know about fungi and mushrooms, and you probably have a picture in your mind when I say mushroom of a thing with a stalk and a cap and gills, but that's really just the smallest aspect of what's out there in terms of the diversity of fungi. And so there's some that I'm quite interested in that are kind of egg-shaped little fungi, and they live underneath the ground and they never sprout out into that classic mushroom shape. And some of these are really, really rare. And so one of the species that we called fish's egg, in Latin, Claustula fisheri, is incredibly rare, and we only found it from the Nelson region. And we found out that the Nelson Council was going to put some mountain biking tracks just through the one locality where we knew this exists. So we spoke to the City Council and they agreed to move the tracks to preserve the habitat of this rare mushroom. And I know it's kind of a strange thing for most people to think. You think rare and endangered and birds and lizards. But yeah, mushrooms are rare as well, and we've put about 50 mushrooms on the rare and endangered list so far.
The fish's egg, can I just ask, how do you find it if it never comes above ground?
It's a great question, and we have very expert scientists who can notice these things, and I don't have my eye in for myself, but I've been out in the field with them, and they look at a little patch of ground and they're like, it's something looks different over there, and they've got a truffle fork, and so they can scratch the ground and find these hypogynous fungi that live underground.
So can you tell us about how the arthropod collection has helped with biodiversity?
Sure. And so the New Zealand arthropod collection, and arthropod might not be a word that people are familiar with, and really this covers all sorts of creepy crawlies like insects, or true insects with 6 legs, and spiders, but also nematodes, these tiny little microscopic worms as well. So the New Zealand arthropod collection is incredible. So we've got more than 1.6 million individual specimens pinned down in drawers, plus quite a few more in ethanol storage as well. And one of the fascinating species that we have there is called Fred the thread. And Fred was found by one of my colleagues, Corinne Watts, when she was looking at plants that grow in these peat bogs of the Waikato region. And she noticed some kind of strange patterning on the leaf and thought that it might be a little insect that lives inside the leaf. And so she dissected it out and found just the craziest little critter in there, and it was very thread-like. So it was a deep kind of orange-red colour, only 1 millimetre thick, but about a centimetre or so long. And nobody she took this organism to could kind of figure out what it was. Nobody had ever seen anything like it before, and the beetle people were like, oh no, this surely not a beetle larvae, and the fly people were like, oh no, this doesn't look like a fly larva. So she reared it, so let it develop, and it turned into a crazy little moth. And so Fred the thread, as he's called, is one of these incredible organisms with a crazy little flip-top head that lives inside a plant that most people wouldn't be aware of. And this is just one of the amazing things about the biodiversity that we have here. And so the NZAC collection was able to look at all of its different moths that it had and found if one matched Fred or not. And we now have that preserved in there. And with all that data associated, we now know where Fred and its kin are all around the country.
Does it live in a certain type of plant?
Yes, it just lives inside a certain plant that grows in these peat bogs called giant cane rush.
Very cool. So further on how these collections have helped with biodiversity, how have some of these other collections been used to identify species richness within New Zealand?
One of the interesting things is that it's not actually that immediately obvious what species we have here. And so, for example, kowhai trees, there's actually quite a few different species. And one of my colleagues, Peter Heenan, about 20 years or so ago, did a lot of work looking at all the kowhai that he could see across the entire country and described a few new species. And this was during the time of my PhD, so I was quite invested in it because I was working on bacteria associated with them. And some of the characters that he was looking at, for example, were what do the little leaf hairs on the bottom of the leaf look like? These kind of characters. And then so he worked across all the species that he could in the collection and did fresh ones, and found a couple of new species, but also kind of interesting discrepancies as well. So one of the species that already existed was called, in Latin again, Sophora chatamica, and as you can tell from the last name, it was described from the Chatham Islands. But he also noticed that this species was present in Wellington in a few little trees, but actually was really common around the Auckland region. And in fact, doing a lot more intensive work, he discovered that actually these trees that were found on the Chatham Islands didn't start there, but they probably started in the Auckland region and then were traded by Māori pre-European colonisation as part of the big trade network that was going on, taking pounamu from the South Island and trading it for North Island species.
How do technologies like DNA sequencing and maybe even a fossil record, how does that help with these studies of species diversity within New Zealand and tracking, which direction it went hundreds and hundreds of years ago?
Yeah, I think taxonomy has embraced the new technologies while also retaining all the old traditional techniques as well. And so when I first started, DNA sequencing was sort of just coming in as an easy to do thing rather than something complicated. And certainly for bacteria and fungi, looking at the DNA sequence is actually really important, because honestly, even though I love bacteria, they all look kind of like gloopy blobs on an agar plate, right? So the advent of DNA sequencing and now genome sequencing is fantastic for pulling out those differences between species and even down to population levels as well. Fossils are still really important though, because they put a date, sort of like a boundary of when that species first arose, and then we can use a combination of dating the fossil through radioisotopes or those kind of methods, and then molecular clocks. And so DNA evolves, changes, mutates, and it does so at a somewhat regular rate. And so we can use that rate of change in DNA as a clock to kind of count back in time when that species first arose or where that speciation event happened.
It's just a wonderful combination of sciences leading to some fascinating stories. Switching to biosecurity now, let's look at some of the stories, the impact stories that have come out of the utilisation of these collections to help with biosecurity controls. So can you tell us the story of the fireweed, the new weed that was killing livestock, and how the collections helped with that?
So New Zealand is a very weedy nation. We've got a lot of weeds that have come in from overseas or escaped from gardens. Some of them are just a nuisance, they look ugly or they take up some space, but some actually are really bad for the New Zealand bioeconomy. And this particular example is called fireweed. It's a senecio plant, and it was first found in 2017. Some unusual samples were noticed in a pasture in Northland, and they sent these samples down to our Allen Herbarium, CHR, down in Christchurch and Lincoln. And our taxonomists there looked at them and found that, this was actually a new species to New Zealand. And fireweed is quite a problem because when livestock ingest it, it's toxic and can lead to liver damage and eventually death. So it's quite an important weed. So our taxonomists were able to identify this and notify MPI to take action on this. And those samples that were taken and stored in the collection were later used for DNA analysis to analyse the entire Senecio genus to look at taxonomic revisions and understand what the species are.
The New Zealand Fungarium collection might just be my favourite collection, I think. How has this collection influenced the bioeconomy and biosecurity efforts?
So the New Zealand Fungarium and each of these collections has a weird acronym, and sometimes it makes sense and sometimes it doesn't. And so here the acronym is PDD, and this stands for the Plant Diseases Division of DSIR, the former organisation. And so the collection has native mushrooms in there as well, but it also has very strong collection of plant pathogens. And following normal good scientific practice, when everyone discovers a new pathogen in the country, they'll write up a scientific publication, of course, but the best practice is to deposit some of those samples in the collections for future use and for validation that you've got the thing right. And so a guy called William Hartle in 1991 found some avocado disease in New Zealand, and it was an avocado scab was the disease name, and so he wrote a little paper on it and deposited the collections in PDD and ICMP, as he should. Fantastic. But then fast forward about 10 years, and we end up that we're exporting avocados to Australia. And the Australian biosecurity people intercepted a shipment and saw some scabby-looking avocados. And they'd stopped all export of avocados to Australia, which obviously, isn't the situation that we want to have. And so we investigated this along with Kerry Everett, one of the pathologists, and we went back into that collection that William Hartle had deposited and re-examined it. And fortunately, we also in the collection have real examples of avocado scab from overseas. And so we compared them by looking at the lesions, so that the scabbiness of them, and found that they didn't really look quite right. They didn't really match real examples from overseas. And then we looked at the culture, so the organism that was isolated from that scab, and found that was a completely different organism, not the avocado scab one. And so these two sort of pieces of evidence were able to put together and establish that avocado scab was not present in New Zealand at all, and that symptoms was probably just a wind rub, so branches causing abrasion on the surface of the fruit. And the Australian biosecurity guys were happy with this, and we were able to restart avocado exports back into Australia.
Wow, that's a wonderful impact story, and so important with New Zealand being an export nation with horticultural crops, that we really look after those pathways. So New Zealand also has a thriving forestry industry, and accompanying that we have the National Forestry Herbarium, which includes indigenous and productive forestry trees. How has this collection been used for biosecurity efforts in New Zealand?
We often think, when we think of forestry, as pine trees, but eucalyptus trees are also actually a very, very important part of the mix of wood that we export. And so particularly in the lower part of the South Island, the eucalyptus species there cover about 10,000 hectares. A few years ago, there was noticed an incursion of a eucalyptus beetle that attacks these trees. And we use the specimens in the National Forestry Herbarium to understand which eucalyptus species were going to be affected by these beetles, and determining the impact on forestry and the export of logs to be able to minimise that impact.
Great. The public would know about insect incursions such as the brown marmorated stink bug. Right now in Auckland we've got the problem with the yellow-legged hornet, and there's a sort of a constant threat with Queensland fruit fly. So how can the collections that we have, particularly the arthropod collection, how can that be used to help battle all of these biosecurity risks?
One of the great things about having so many specimens is that we have different variations of each species. And so if we take photos of all of our different kind of variations of the form, we can use that with some sort of automated species identification to be able to take the burden off people doing the identifications. And we tried this with brown marmorated stink bug, and triaging thousands and thousands of surveillance photos, and discovered that they were able to, automated tools were able to identify them about 93% of the time accurately, which is fantastic if there's a big outbreak or response. We did similar work with fruit flies, which are also, of course, a big pest, and in this project found that 98% of the identifications were right. So not perfect, but fantastic at taking the workload off people.
Absolutely. Although not perfect, probably maybe better than the human eye sometimes. Yeah, that's great. So, yeah, I guess we talked about whole genome sequencing before, and you've mentioned, computer vision, deep learning now for high throughput phenotyping. And people often present that question of, which one do we go forward with in like an either or sort of sense. But so just spinning that around and thinking more like collectively of a both and, kind of using all tools in the toolbox, how do you see advanced technologies enhancing the utility of our nationally significant collections and databases?
I think it's a plus. I think all these new technologies have been incorporated over the time. Like I said, taxonomy has been around for 300 years, and we just absorb all the new technologies as they come in and use it as best as we can. And so genome sequencing has been fantastic to be able to identify cryptic species that can't otherwise be analysed. And it adds another layer. So for microbes, for example, yes, we can use those genomes to identify them, but then we can also look at their genes that control their ability to cause disease or to make toxins.
So all throughout this, I've been considering how important it is to have expertise and taxonomists and biologists working together, but also the people out in the field spotting things like fireweed and unusual plants and insects. So why is the expertise of taxonomists and biologists so fundamental to the efficacy and impact that these collections and databases can have?
New technology is fantastic. It's really able to enhance what we do, but technology doesn't have curiosity and technology doesn't have passion. So Fred the thread would never have been noticed by an AI. It was just that passion of the scientist noting something weird in the plant and then going to the effort of rearing it up and noticing such a crazy, unusual organism in there. We will always need people and their incredible passion and experience.
And curiosity, like you say, for sure. So speaking of people and how people can work together, perhaps more seamless interaction, what do you see are the main benefits of having a lot of the taxonomists, biologists and other expertise all together now under the Bioeconomy Science Institute banner?
I think it's a fantastic opportunity. My most favourite projects that I've had over the past 20 years is when we're working in that multidisciplinary space. And so I've recently worked on the facial eczema disease of cattle and sheep, and was bringing together, me and my team as taxonomists, fungal genetics people, understanding what's making the toxin, pasture people, understanding how pasture grows, what's on there, and the animal biologists. And bringing all these people together in a single institute, I think is going to be fantastic for future impactful research.
Yes, it's a bright future for sure. Lastly, considering the public and community and the utility and access to these collections, how does the public interact, or how can they, or do they? How do we get public and communities making the most of these collections as well?
So part of the funding that we receive for these nationally significant collections is to make them available to all researchers and the public as well. So researchers can actually borrow the specimens and do work on them, but for the public we have some public-facing websites which people can look at to determine the best name to use, because taxonomic names change all the time. And also looking at photos of the different organisms, and we've got a tool that will enable you to, for example, identify the curious little bug that you found in your backyard.
Very nice. That's great. And as a little plug for that tool to identify curious-looking bugs in the backyard, where do people go?
You can just Google or search for "what is this bug?"
Beautiful. Lovely. Thank you. Well, it has been an absolute pleasure talking with you today and learning about the impact of these nationally significant collections and databases. We really appreciate your time and expertise. Thank you for being on the podcast.
Thanks for having me.
And thank you out there for listening. If you'd like to know more about the topics covered on SciGest, check out the episode pages on the Plant and Food Research website. Please also subscribe to or follow the SciGest channel and rate and review each episode. Thank you and mā te wā.